Morning. So today we will finish talking about membrane transport. Remember, we've been discussing for the last couple lectures the importance of our membranes in our cells, both for defining the outer cell membrane that holds all of the sort of guts of the cell together, and membranes that make up different structures inside, things like organelles. What we've been talking about is how important it is that these membranes, which are made of a bilayer of phospholipids, is not like a solid structure, right? And in fact is very fluid because it's just made of these layers of phospholipids that just come together because water is surrounding everything. And the water tends to play nicely with the hydrophilic heads of the phospholipid molecules, and the water doesn't play nicely with the fatty acid tails of the phospholipid molecule. So as a result, the tails just kind of get pushed around, and the natural place that they go is all these phospholipid molecules will have their tails pointed inwards, and that's how you get the bilayer. And that bilayer then is embedded with different types of proteins that serve functions, including regulating what things go in and out of the cell through that membrane. So it's important to remember that in living things, our phospholipid bilayers are semi-permeable, right? We control, or our cells control, what can go in and what goes out, and that careful, finely tuned level of control is essential in order for all of the biochemical reactions and things happening inside the cell to happen in the way that we need them to. Last lecture, we looked at diffusion, the sort of concept of things go from a high concentration across a membrane to a low concentration in order to even things out. And we began to explore this idea that you can think about concentration gradients as being numbers of molecules. So a high number of molecules on one side and starting with a low number of molecules on the other side, how they will want to just even out. But we also have to think about charges, pluses and minuses, also want to evenly distribute by going back and forth. So diffusion, simple diffusion, we said was just when molecules are allowed to go through the membrane. The membrane slows them down a bit, but over time, if things are simply diffusing, right, things will even out. In some cases, there are molecules that cannot get through the membrane because they're too big or the way that they're charged. And as a result, you need to have different types of specialized proteins embedded in the membrane that will allow these particular biological molecules to go in and out as needed. So we said if the moment you start involving proteins to facilitate this process diffusion, then we just call it facilitated diffusion. And remember, those instances of facilitated diffusion do not require the cell to put any extra energy into this process, right? Other than the making of the protein itself originally, right? But then the protein is just designed to allow things to flow along their natural high to low gradient, right? So that was simple diffusion and facilitated diffusion. Today, we're going to take a look at active transport. In other words, this is no longer diffusion, because what we're talking about now are those many instances when cells need to actually make things go against their gradient, move things across a membrane from a low concentration to a high concentration. And that's going to require energy to do so. So let's take a look at this. Let's start off to get on the same page again by thinking about the effects of water as a solute on different things, that water as a solvent, sorry, the effect of it on solutes that are dissolved in it, right? You know, over and over again this semester, I always emphasize, I want you guys to think about how important water is for how life ended up being the way it is on planet Earth, right? So I think any examples that help us think about the importance of water are both insightful and I think a lot of times really interesting, because we oftentimes don't think about things this way. Let's say you have a glass of water and you dissolve a teaspoon of salt, sodium chloride, in it, right? So obviously you have salt crystals that are a solid, and you put it in the water, you swirl it around or stir it around, and eventually what? The crystals disappear and the sodium chloride is now dissolved, as we say, into solution. Right? So if you look at a NaCl crystal, remember the bonds between the Na ions and the Cl ions are ionic bonds. These are not covalent bonds. In this case, the sodium ion has given up an electron, right, to become an ion. Right? So it becomes Na plus, and that electron is taken by the chlorine to become a chloride ion with a extra electron, so it's negatively charged. Right? So they are not sharing an electron, but since the sodium becomes positively charged and the chlorine becomes negatively charged right after the electron is donated, then the sodium just happily will naturally want to kind of hang out there, right? Because its positive charge is just attracted to the closest negative charge, which is this chlorine. So that's why the spaces between the Na plus and the Cl minus ions, right? Those are actually, there's no physical sharing of any electron clouds, no strong bond there. Those are spaces in between which water molecules could get. Right? So when you drop this crystal into there, what happens is it's surrounded by water, and you can see the individual water molecules just kind of like get in between. And because the water is polar, right, there are water molecules that will happily play with or surround the positively charged sodium here. So the red oxygens, right, are attracted to the positively charged sodium, and the water molecules will also happily play with or surround the negatively charged chloride ions. But in this case, it's the hydrogens, right, the protons on the waters that encircle them. So you can really imagine that if this thing were animated, unfortunately it's not animated, but you can imagine the water molecules as they're bumping around in here, eventually they peel off, right, the sodium and the chloride ions, and that's how the sodium chloride salt crystal dissolves in water. So this is because it's highly hydrophilic. The opposite would be, let's just pretend, for example, that this was a droplet not of purple and green sodium chloride ions, but these were just droplets of a droplet of fat, right? So these balls would be individual fatty acids, right? That might be negatively charged. Well, not negatively charged. They would be hydrophobic, right? Because they would be more or less charge neutral. So the hydrocarbon tails, right, would not play nicely with the water. And as a result, the water wouldn't really be getting in between and breaking up this oil droplet. Instead, it would just keep knocking around on the outside of it, right? And any of the little oil molecules that might want to float out, they would just naturally get just shoved back in again, right? Because the water molecules surrounding it would just want to put all of these fat molecules into the most sort of limited space it can, so that all the water molecules around it happily have their space, right? So in that case, that's why an oil droplet stays together. All right. So let's talk about this concept of when you dissolve something like salt in water, what are some of the terms that we use and think about here? So osmolarity would be the total solute concentration. So you'd say the osmolarity of this solution would increase with more salt that you dissolve and add in there. Right? At a certain point, right, you'll put in so much salt that there's just no more space among all the water molecules to allow the sodium and chloride ions to come apart like this, right? In which case the solution would be saturated. Osmotic pressure or osmotic potential is the tendency of the water molecules here to be encouraged to flow through a semi-permeable membrane to allow salt solutions like this to equalize on both sides of that membrane. Let's illustrate another concept here, right? Instead of just talking about salt crystals, let's talk about something that we have been spending a lot of time thinking about, and that is proteins. So we've already learned that many proteins form complex three-dimensional shapes as a function of their amino acid sequence, right? Their amino acid sequence that represents a chain of amino acid residues with different R groups sticking off of them. Remember, we have 20 different amino acids that are needed for life on Earth. right? Some of them have long polar chains, some of them have small non-polar chains, some of them are charged, some of them are not charged. When you have a protein that folds up in 3 dimensions, right, how does the different R groups sticking off of these amino acids affect the shape of the protein? As you remember, the eventual shape of the protein, right, is the key to whether or not that protein will be able to function and do its job. Right? For example, interacting with some other molecule that is the right shape to bind to it. Here, let's think about this model on the left. The model on the left shows an overlapping of the peptide backbone, the polypeptide chains, that's the dark blue, with these secondary structures like helical structures and sort of unordered looping regions. And that's how this chain will fold up. The lighter blue sort of clouds around them are the electron clouds as the result of the different R groups of the amino acids sticking out from that central polypeptide chain, right? And that forms the 3-dimensional shape. If you were to interrogate or kind of survey each of the amino acids that makes up this chain and figure out if it's polar, non-polar, so therefore hydrophobic or hydrophilic, this is what you would find. So the hydrophobic residues, in other words, the amino acids that have hydrophobic R groups, they have their R groups pointed towards the inside of the protein, whereas the hydrophilic R groups tend to be on the outside. Right? Why is that? This is entirely because this protein in real life is not floating on a white background PowerPoint slide, right? It is floating in your cell, surrounded by lots of water molecules. And those water molecules, right, will play nicely with the hydrophilic R groups, the tails of those amino acids. So they will tend to kind of like encourage or pull those towards the outside, right? But any of the hydrophobic tails of amino acids, the water molecules, you know, they don't play with them nicely. They just bang into them. And so that eventually just encourages them to end up being folded towards the inside, right? In other words, yes, right, we've talked about how the amino acid sequence of a protein determines in large part its 3-dimensional tertiary structure, right? But what we really want to appreciate is the reason why that works is because you have water molecules surrounding it. If it weren't for the water, right, this protein would not fold up like this. It would just be this big mess, right? A big, long, loosely disordered peptide chain. Look at this animation here, right? So this helps you appreciate the fact that it is surrounded by water molecules, right? So all of these amino acid residues of the protein that are hydrophilic are being kind of teased out by the water molecules because they're all sort of binding with them, right? Whereas any hydrophobic R group that tends to kind of start coming out, the water molecules would just push it in. And so as a result, the protein, right, is not a solid object. It's this kind of itself fluid chain that's been wrapped up, right? And it's just kind of like on an atomic scale, kind of moving around, but it's held, right, more or less in this same shape because of the water around it. Okay. Osmotic pressure. This was something that when I was, a college student, I always had some trouble with this because to me, this sounded sort of counterintuitive. Let's pretend you have a solution of whatever this orange ball is here dissolved in water. Right? Water is the little blue molecules. So the orange balls are the solute and the little blue molecules, the water is the solvent. Right? And let's say you have these two sides inside this chamber divided by a semi-permeable membrane that has holes in it, holes that are just big enough for the little water molecules to flow through, but too small for the larger orange balls to flow through. Right? Over time, as you let these molecules kind of settle, what you find is that the water molecules will tend to go from the left side, right, the lower solute concentration side, over to the right side. Right? This would be the flow of the water molecules. This would be their downhill gradient from high to low. Right? So I think what messed me up when I was a student was I kind of thought that intuitively, shouldn't it be the other way, right? I always thought like, well, if you have way more orange molecules on the right side, wouldn't that be kind of like squeezing out the water? And so the water would want to go like to the left? No, right? Because the way to think about this is that both sides want to even out in terms of total numbers of orange and blue being the same on either side, right? But because this membrane only allows the small blue ones to go through, the orange ones cannot. So they won't be able to even out, but that doesn't stop the little blue water molecules from evening out. When you think about it that way, then it makes more sense, right? Because at the end of the day, then the number of water molecules on either side, at least, will even out, right? The key to keep in mind here, right, is that will only happen if there is enough physical space on the right side so that you're actually allowing more molecules to go on that side, right? So if the volume on that side is allowed to get bigger, then that's what will happen. But what happens if you confine, right, the volume and don't allow for extra space for those extra water molecules to go through, right? If we're talking about like a cell that's surrounded by a membrane, this can have a big impact on what goes on with the cell and its normal physiology. So diffusion of water is what we call osmosis, right? So osmosis is just a specific case where something is diffusing and that something is water. So controlling osmosis then is vital for cell survival. So here's a sort of classic example of what can happen if you mess with the osmotic potential surrounding cells. Here we have red blood cells, right? So red blood cells are obviously surrounded by a cell membrane. So that's the semi-permeable membrane that we were talking about here. In normal healthy tissues, right, the solute concentration or the tonicity of the surrounding fluid, right, is carefully maintained so that your red blood cells and other cells in your body for that matter are happily in balance, right? The water flowing in and out of them, right, is carefully controlled and regulated, and there isn't like a huge movement of water in one direction or the other. So that what we would call the red blood cell is placed in an isotonic solution, right? Iso kind of meaning, right, the same, right? What if you took those red blood cells and put them in a much more hypertonic solution? In other words, a solution that has a lot more salt in it, right? Then just like on the previous slide, the water from inside the cells would want to seep out in order to even out the number of water molecules on either side. So in that case, if the water's leaving the inside of the cell, your poor red blood cells would shrivel up, right? And they would not be able to function properly. The opposite would be, what if instead of a normal sort of tissue fluid environment, you took those red blood cells and you put them in pure water that has nothing else dissolved in it, right? Therefore, relatively speaking, the concentration of salts would be much higher in the cell than outside, and the water from outside would rush into the cell. And then the red blood cell, right, in order to accommodate that, the volume has to increase. But unfortunately, this can't go on forever, right? At a certain point, this poor red blood cell will simply burst. Okay. So that's kind of, I don't know, a little scary to think about, that all of the cells in your body are in this constant, sort of heavily regulated isotonic type of situation where they're allowed to be happily existing and carrying out various functions. So let's take a closer look at the composition of the different fluids inside of our living tissues. Right? So one of the things that contributes greatly to the tonicity of our fluids is the amount of different ions that are dissolved in there. You know, and by ions, I just mean in a general sense, charged molecules. Right? If we think about our body and we think about all the fluid spaces in our body, you can, in a very sort of generalized macro way, think about just three different sort of compartments in your body, right? you have fluid inside of all of your cells that are contained by the cell's cell membrane. Right? That would be your intracellular fluid, right? So all of the fluid inside of your cells you can think of as one sort of functional compartment of your body, right? Then you would have the fluid that's immediately surrounding the cells as they are part of tissues. Right? This would be the interstitial fluid. So in other words, the fluid that's just between the cells that make up your tissues. And then you have the fluid that's flowing through your circulatory system, right? And that would be mainly all the water that's in your plasma, blood plasma. So those are sort of the three main fluid compartments inside of our body. And you might be surprised, or maybe not, that the balance of different types of ions in these three compartments varies sometimes very greatly. Let's just for simplicity's sake, because we're talking about membranes here, right? The easiest example is to think, well, intracellular fluid is inside of the cell, and then right across its cell membrane on the outside would be the interstitial fluid. In both of these fluids alone, you'd have a balance of plus and minus charges, right? Because naturally, that's how they would be driven. Right? In a fluid, you'd naturally, over time, have the constant balance of plus and minus, but the relative number of different types of plus and minus ions that are involved on either side varies greatly. I think most notably, look at this. Inside of your cells, the intracellular fluid, you have a lot of potassium, but relatively little sodium, right? Compared to just on the other side of the cell membrane, outside of the cell in the interstitial fluid, it's the opposite. You have high levels of sodium and low levels of potassium, right? And so this clearly is not allowing the sodium and the potassium to even out naturally across either side of the membrane. Clearly, there is some effort being made here, right, to keep one side very high in sodium, one side very high in potassium. Right? So how then do our cells maintain this? So, right, the theme of today's lecture is active transport of molecules. What is happening is we have these protein pumps on the surface of our cells that function to maintain on purpose this disequilibrium of sodium and potassium across the membrane. So this particular example here is this protein complex that we call the sodium-potassium pump. Right? In this illustration, you can see that it's depicting on the inside of the cell, we're looking at 3 sodium ions. On the outside of the cell, we're looking at 2 potassium ions. Keep in mind that you could fill in the rest of this and actually show that outside of the cell, right, in this light blue space, that's the interstitial fluid, right? You already have a ton of sodium, right? So you might have many little gray sodiums already here in the picture if you wanted to draw it that way. Right? Likewise, right, on the inside of the cell, the intracellular fluid, you have high levels of potassium. So to start this picture off, you might have many green balls of potassium already in here to start with. Right? What does that mean? That means if you have many, many more gray sodium balls on the outside here, then what would be the direction that the sodium ions would actually naturally want to flow from uphill to downhill? It would be from out to in, right? And likewise, the green balls, if you start off with way more of them inside the cell, right, the natural flow would be the potassiums would want to go out. So in order to maintain on purpose this imbalance and keep that imbalance going, the cell has to actively pump these ions back and forth against their natural gradient flow. Right? So you can see here, that's what this cartoon is showing. This antiporter, we call it an antiporter because it's going to transport 2 things in opposite directions, is going to take 3 sodium ions from the inside, load it up, and it's going to move them out. And in the process of doing that, it's going to take 2 potassium ions from the outside and move them in. In both cases, right, going against the natural flow of where these green balls and gray balls would want to go. Let's look at this, how this works. So here we show at the start, we have that sodium-potassium antiporter in its starting state. It binds to ATP, and that ATP is going to provide the energy for this thing to be able to go against its gradient. Why does it need energy, right? I mean, yeah, you all understand, like, yeah, it's going uphill in the sense that it's taking something from low concentration to high concentration. But where does the energy part actually fit into this, right? I mean, this is not like a motor that you plug into your outlet or put some gas in a engine or something like that, right? In that case, you're like, oh yeah, right, it burns that energy. This is molecular energy in the form of ATP and a high energy phosphate bond from that ATP. That's going to provide the energy. If you think about the antiporter protein, right? So the protein complex is made up of these purple alpha units, this navy blue gamma unit, and this orange beta unit. So again, right, it's a quaternary structure of different proteins working together, right? You don't have to know this alpha, gamma, beta, but you need to sort of get this concept, right? That this structure, it's floating in the membrane. And again, right, it's kind of fluid, right? Because it all has to do with the water molecules surrounding it, right? And so it's kind of this fluid shape, but it's in a nice shape where it'll bind ATP. And by binding ATP, that kind of encourages it to stay in this kind of starting position here, right? And in this starting position, 3 sodium ions, right, will fit very nicely into this specially shaped pocket here. At that point, when the sodium ions bind, it kind of nudges the proteins around, and it nudges the proteins around enough that it causes the ATP to be broken. And that third phosphate bond on the ATP is broken off, and that phosphate, the third phosphate, reforms a bond with the antiporter protein, right? The purple part here, okay? And so therefore the ATP is converted to ADP, which just floats away. So now you have this phosphate group stuck to it, to the protein, right? So this protein has been phosphorylated. As a result, that particular event causes the peptide chain of the protein to kind of have to like readjust itself, right? And assume another more comfortable position now that it has a phosphate group stuck to it, right? And that more comfortable position tends to mean that this side of the protein, the outside, opens up. And these sodium ions then naturally, because water then, right, there's water molecules here, the water molecules surround the sodium ions, just like the dissolving table salt in the water, and they carry the sodium ions out to the outside. So when this happens, the new shape that's shown here, right, is the right shape for the sodium to come in. Not the sodium, sorry, the potassium to be loaded in. Right? And when the potassium's loaded in, that then kind of changes the shape of the protein a little bit, and that causes the phosphate group to fall off, right? And then as a result, the potassium ions are happily brought into the cell. And then in this starting shape, ATP then happily binds to this again, and this process keeps going. So in other words, the energy is in the form, right, of actually adding a bond here to the protein so that it forces the protein to change its shape, right? And in other words, that amount of force by forcing a phosphate group onto this protein, right? That amount is enough to be stronger than the force of the potassium or the sodium ions wanting to resist, right? And float in the direction that they normally would want to float in. That's an interesting way of thinking about it. So active transport then is always movement of some sort of biological molecule against its natural gradient flow. Right? And this is really important because there are many situations where you have cells that might be, filled with different nutrients that it's busy using to make things like macromolecules and things like that, right? And so the concentration of certain types of nutrients inside the cell might already be much higher than the availability of those nutrients outside of the cell, and yet the cell still wants to be greedy and take more in, right? So nutrients flowing from the low outside to the high inside certainly would require energy because that's going uphill. Secreting products and wastes, right? Oftentimes, the cell is producing waste products that it needs to get rid of, but in your tissues surrounding the cell, because there's like another billion or so neighboring cells all sort of putting waste out into the interstitial fluid, maybe the waste concentration is already high on the outside, but yet the cell still has to move the waste out against that, right? So that requires energy. And as we just looked at, maintaining intentional disequilibrium of certain ions like potassium, sodium, calcium is another really important one, and protons, H pluses, right, in order to maintain disequilibrium, right? That also requires energy. So you might say, like, well, why though, right? Why does the cell care to on purpose keep sodium levels and potassium levels so out of whack and so intentionally imbalanced, right? It's because oftentimes what the cells are trying to do is create gradients that it can take advantage of later. And we'll look at that in a couple slides. Important thing to keep in mind, anytime you're moving something against this gradient, it's going to require energy to move things uphill. Right? So let's look at this example here. Active transport, right, requires energy, but it doesn't necessarily mean that that energy is in the form of something directly, binding to a protein and forcing it to change shape. It can, and of course it often does, but it doesn't necessarily mean that proteins are designed that way. Right? So this is a comparison of direct versus indirect active transport. So here we have two examples of two different transporters. So these are two different proteins. Okay. The one on the left is an example of a direct active transporter. It's kind of the easiest, most intuitive one to think about. Here we have, just like with the sodium potassium pump that we looked at before, right? This protein is the right shape to bind something like ATP, and it causes the breaking of that high energy third phosphate bond. And that phosphate bond, then this phosphate group could either end up being added to this protein to kind of force it to change its shape, right? But it doesn't have to. It could just be that the involvement of breaking ATP is enough to change the shape of this. In this particular case, right, because it's directly using ATP, that's why we call it a direct transporter. And you can see it is moving protons, right? It is moving protons from a low intracellular concentration of protons, pushing them uphill, right, against the natural gradient, because on the outside, you have a lot of protons. An indirect active transporter, like this purple one on the right, look what it's doing, right? What it is doing, it is taking advantage of that artificially maintained proton disequilibrium from the outside to the inside and allowing the protons to flow in the direction that they would naturally want to flow, right? If they were allowed to go from high to low, they go through this particular protein. But look at this. By allowing the protons to naturally flow back downhill, that is coupled to the movement of some solute molecule, this red S. And this red S is at low concentration on the outside, and it's already much higher on the inside. So it is being moved against the natural gradient that the S molecule wants to flow in. Right? So what's important to keep in mind here? That means, that the S molecule is being pushed uphill as the protons are being allowed to flow downhill. So the relative force, right, or power of the protons flowing is stronger than the flow of the solute molecule would be if the molecule was allowed to flow from in to out, right? That's how it's able to overcome the gradient resistance of the solute molecule and actually move that against its gradient. So you might look at this and you might say, well, I don't see where energy is used here, right? Because there's no ATP. This is the thing to keep in mind, right? This protein on the right would not work if it were not for what? If it were not for the fact that there is a proton gradient to take advantage of. And where does that proton gradient come from? It requires the direct active transporter on the left that's using up energy to make that gradient. Right? So you might say, well, that's kind of weird, because why wouldn't we just evolve to have all pumps just themselves use ATP, right? Because that's not a very efficient way to do it. If you think about it, if you, instead of just having this one indirect active transporter here on this illustration, you had maybe like 10 more examples of different transporters, right? And if all of them needed to use ATP directly, that means in order for one sort of cycle of this to happen, right, you would need like 10 ATPs used up. Right? Instead, think about how elegant this is. These multiple different transporters have just evolved to take advantage of the flow of protons, for example. So that way you only need to have a few of these proteins actually using up ATP in order to maintain that disequilibrium of protons, and then many other proteins like the one on the right can just take advantage of that, right, without them directly using up ATP. Right? So that's why we call it an indirect active transporter, because technically it does use energy, just not directly. Right? It relies upon some other protein to break the ATP. So many direct active transporter pumps are what we call ATPases, right? So they break ATP, so they are an ATPase. So just like this illustration here, right? This was our one from the last slide. ATP is broken to provide energy that may or may not be the liberated phosphate group being added onto the protein itself. So if you look at this example, right, let's look at something like a jack that's used to raise up a car to change a tire. We can think of a jack as a pretty simple machine. Right? It has a long lever where you can push down on it, right? And then there's like a little pivot here. And then, of course, you have the actual part, the little platform that will raise up the car. Right? So in this case, if we think about this as a simple machine, this is where the energy input is. Right? And then this is sort of where the work that's being done takes place. And in this case, if we were to imagine that the energy is in the form of ATP being broken to provide an extra phosphate group, if that phosphate group gets added to this as part of changing its shape, changing the shape of the jack, right? This would be an example of what we call a P-type ATPase. So the transporter protein itself is phosphorylated in order to make it change its shape, and so it can do its work. So you might say, okay, that's fine, but you know, why do we call these things ATPases, right? Because it seems to be emphasizing the wrong thing. I mean, who cares that they break ATP, right? Shouldn't we just call this, for example, on the left, a hydrogen or a proton pumpase or something like that, right? I want you to think about this, right? If you look at the car jack example here, right? Would this jack work? Would it still work if there was no car in the picture? And the answer is yes, right? You could still put in energy to have the platform raise up, even if you didn't have anything on it, right? So if you think about this, when you design a jack like this, right, the design is all in just simply making the jack move in a certain way. How you use that change in shape is up to you. You know, if it weren't a truck in this picture, but let's say this guy, he has like a porch or something that kind of collapsed a little bit, and he needs to kind of raise up the wooden beams just so he can kind of get in there and put in some other, repairs. He could use this also, right? So that's why you wouldn't call this a, truck jack ace or something like that, right? If you used it to lift up some fallen timbers of your porch, right? So the point is, these proteins, like the one on the left, right, that evolved to do things like move protons, that's almost like a secondary function of it. What it truly evolved to do was just change shape in a particular way in response to ATP being broken and making use of that energy. Right? That, if you think about it, that is an amazing concept, right? Because it's not necessarily the fact that that living things needed to pump protons, and therefore life had to come up with something like this protein. No, this protein first evolved to break ATP. And over evolutionary time, the fact that it changes shape when it binds ATP and breaks it turned out to be useful for different types of functions like moving protons. So that's why we just call them ATPases, right? It sounds like a very generic, boring kind of name, but that actually emphasizes what these proteins really evolved to do, is just to break ATP and change shape. There are 4 types that are really important in our bodies. They're the P-type ATPases. So P stands for they get phosphorylated as a result of breaking the ATP. Other types, like the V-types, are the ones that actually take care of pumping the protons. That's why on the previous cartoon, when the cartoon shows that purple proton pump breaking an ATP, it actually shows the inorganic phosphate just kind of hanging out, right? It doesn't necessarily show it put on there. So V-type ATPases, the V stands for vacuoles, because in many vacuoles in our cells, the proton disequilibrium is tremendous from one side of a membrane of a vacuole to the other. And these V-type ATPases take care of that. There are F-type ATPases that are also proton moving or transporters, but these are coupled to things such as making more ATP molecules. So we will study those later on when we look at cellular respiration. And the 4th type are these ABC type ATPases, which ABC just means ATP binding cassette. Many of these are used to transport small inorganic molecules out of the cell. So for example, different types of poisons or medications or drugs that get into the cell, right? The cell needs to kind of get rid of those. So many of these ABC type ATPases use energy to pump them out. All right, so let's just kind of reinvestigate this idea of the indirect active transport, because I think this throws a lot of students off, right? So remember, this one was the indirect transporter in that cartoon we looked at before. It uses a proton gradient, right, in order to power moving a solute against the solute's natural gradient flow, right? And we said it's confusing because we don't see where the energy actually is used up. Why is this active transport, right? Look at this example of this water mill, right? So I think in this water mill here, I think we all can intuitively understand that if you have water flowing down over the water wheel, the mill can be turned, right? And inside this little shack, you can be doing some sort of function, right? There's gears and stuff that will turn. If the water doesn't actually flow over the wheel, but instead just flowed under it, the wheel is not going to work, right? You're not going to be able to get any work done. So this requires that the water is elevated to start with. So in this picture here, right, how did the water get elevated? Oh, yeah, obviously somewhere else, there must be some sort of a pump or something pumping the water up so that it can be allowed to then fall down. Right? So imagine if you have somewhere this big pump that pumps the water up, and then there's a whole network of elevated waterways, you know, that are running all over an area. And then you have many, many little wheelhouses like this, right? Each that can then take advantage of its own little waterfall. Right? So that's the concept. Energy was required at some point to get the water up so that it could naturally flow down, and that energy of the water flowing down is then harnessed to do work. Right? So this is still considered active transport, just not directly. All right. So let's look at an example of one of these indirect active transporter. We had previously looked at glucose transport from outside to inside as being facilitated diffusion. In other words, if you have a situation where you have a cell and there's high amounts of glucose on the outside and low glucose on the inside, then naturally the glucose will want to flow into the cell across the membrane, but glucose is too big to easily go through the membrane itself, so you'd have different proteins that facilitate or allow the glucose to move easier through little openings in the membrane. So the GLUT1 transporter was a good example of that. There are certain cells, for example, the cells of your liver, whose job it is to be like real hogs of sugars, right? So glucose gets taken up by your liver cells and concentrated inside of the cells so they can make things like glycogen. So in that case, you have a situation where the concentration of glucose inside of the cell is already much higher than the glucose available outside. So if the cell wants to be greedy and keep moving glucose in, it's going to require active transport of glucose. So here, this purple shape is the sodium glucose symporter. Again, think of it as being this kind of fluid shape, right? That's kind of moving around, right? But if it happens to be in this kind of open configuration, if you have sodium ions from the outside, those sodium ions will bind to it, and that will make it the right shape for one molecule of glucose to also fit in there from the outside. And when those three molecules bind on the inside, oh boy, right? The purple protein parts, it has to kind of shift around because it's not too happy anymore. And so the shape changes so that it opens to the inside. So the sodium ions fall into the cell, right? And importantly, the glucose also falls into the cell. Right? When I say falls, what do I mean? Water comes in and takes these things out, right? And so they naturally just float into the inside of the cell. And then the protein then resumes its kind of fluid shape again, and this process starts again. Right? The important thing to keep in mind, right, is that these sodium ions, they have to be pumped back out of the cell in order to maintain that high extracellular concentration of sodium that allows them to like flow back in step 1, right? So this step at step 4, not pictured here, would be some other active transporter that would directly use ATP to pump the sodium out. For example, right, the one that we looked at earlier in this lecture, the sodium-potassium antiporter, right? So that's how that would be pumping the sodium out so that this sodium glucose symporter can work indirectly. But technically speaking, this still requires energy. Okay, last slide for today. This is kind of a fun slide. I mentioned V-type ATPases as another example of these ATP breaking proteins that function to move something. In the case of V-type, they move protons. Right? So these V-type ATPases are very effective. They can build up proton gradients, right, of high H plus on one side of the membrane and low H plus on the other side, about 10,000 times, difference, right? That's amazing. And so this is often used in vacuoles in our cells. So that's why they're called V-type. So if you think about a gumball machine, for example, and the gumballs are like the protons, let's say, right? So you need to put in some energy here to spin the crank on the gumball machine, right? And that will mechanically move 1 or 2 gumballs, usually 1, but if you get 2, hey, you're lucky, right? Through it. All right. So I think we all understand that, right? Energy is needed to crank this, to move some parts that then physically move our gumballs through the gumball machine, right? So why do I use a gumball machine example? Look at the structure of one of these V-type ATPases. Here it is, right? I think this is wonderful, right? So this is a complex quaternary structure of different protein subunits, right, that form a little machine, right? But in this case, the little machine is made up of individual parts, right? And what happens is the way this is designed, these different subunits work together so that the binding of ATP and the breaking of ATP into ADP causes the central rotation of this bottom part of protein subunits to spin and go around. Again, just because of the shape of the proteins changing so that they end up moving in one direction around and around. That is the mechanical movement that can then move these gumballs or these protons from one side of the membrane to the other, right? And you might say like, well, this is just a cartoon then. It looks like a gumball machine. That's just a cartoon. What does it look like in real life, right? Here you go, right? Isn't this fantastic, right? At this point in the semester, right, we are comfortable with this idea that a big quaternary structure like this is made of different protein subunits, these A1s, these B1s, C, D, E, F, G, right? Each one of these, remember, right, is a separate protein that is encoded for by one of your genes in your genome. Right? And that gene gets transcribed mRNA is edited from it to make the finished mRNA that gets translated to make the A protein or the B protein or the C. And then these are folded in just the right way, right, because of interactions of its amino acid side chains with water molecules, so that these things can come together like this to form an undeniably complicated machine, right? And how amazing it is to think this is what's on the surface of your cell membranes right now, right? Doing their job, spinning around and pumping protons. Okay. All right, let's go ahead and stop there today. All right. So unfortunately, I don't have time to answer questions right now. If you have any questions, feel free to email me. And reminder, you have a homework assignment due in recitation this week, and you will have a quiz. The quiz is on the gene expression stuff that we've been talking about sort of last week. So good luck with that, and I will see all of you on Friday. Okay? All right, bye-bye.